We have discussed the two types of immune responses, the humoral immune response and the cell-mediated immune response in our video on innate and adaptive immunity. These two responses are part of adaptive or acquired immunity. Adaptive immune responses require time to develop and are only developed after exposure to a particular pathogen. The adaptive immune response is slower than the innate response. Adaptive immunity is also known as acquired immunity. It is specific, has memory, and acts against pathogens with help of lymphocytes. Adaptive immunity is an immunity that occurs after exposure to an antigen, either from a pathogen or a vaccination. In this video, we will cover some of the basics, such as how lymphocytes are produced. How do T cells work? What is a cell-mediated response? And much more. First, let's recall what we know about the adaptive immune response. The adaptive immune response is brought about by the stimulation of immune cells, activation of cytotoxic T cells, and as well as B lymphocyte stimulation. You may recall we talked about that not all adaptive immunity involves antibodies. Protection against many important pathogens is mediated by T lymphocytes along with phagocytic cells, and B cells play no part. This type of immunity is often loosely termed cell-mediated immunity, or CMI, because historically it was not possible to transfer this immunity from one animal to another, simply by transferring plasma-containing antibodies. Let's get into some more details. Cell-mediated immunity, CMI, is an immune response that does not involve antibodies, but rather involves 1. Activating antigen-specific cytotoxic T lymphocytes, CTLs, that can destroy body cells displaying epitopes of foreign antigen on their surfaces, such as virus-infected cells, cells with intracellular bacteria, and cancer cells displaying tumor antigens. 2. Activating macrophages and NK cells, enabling them to destroy intracellular pathogens. And 3. Stimulating cells to secrete a variety of cytokines that influence the function of other cells involved in adaptive immune responses and innate immune responses. Cell-mediated immunity is directed primarily by microbes that survive in phagocytes and microbes that infect non-phagocytic cells. It is most effective in destroying virus-infected cells, intracellular bacteria, and cancers. It also plays a major role in delayed transplant rejection. The adaptive response is activated when phagocytic cell, such as macrophages, engulfs a pathogen. After the macrophages have engulfed the pathogen, it converts into an antigen-presenting cell. The newly formed antigen-presenting macrophages are then recognized by a helper T cell. The process of T cell activation requires recognizing of antigen that is associated with molecules on the surface of cells called major histocompatibility complex, MHC, molecules. Let's look at T cell activation in more detail. Naive T cells differentiate into helper T cells and cytotoxic T cells once it has been activated. The series of events that bring about this change are as follows. T cells are produced by a process known as lymphopoiesis. Cells known as hematopoietic stem cells mature into common lymphoid progenitor cells, which then becomes either a B cell or T cell. To become a T cell, it migrates to the thymus and becomes a thymocyte, which then differentiates into a mature T cell, or the naive T cell as mentioned before. The naive T cell undergoes a process called priming to change into effector T cells once it encounters an antigen in the lymph node. Priming is based on two types of signals, antigen binding and co-stimulation. The antigen is normally presented on the MHC molecule of an antigen-presenting cell, such as a macrophage or a dendritic cell. It binds with the T cell receptor via epitope. MHC in humans is called human leukocyte antigens, and its proteins are encoded by MHC genes found on chromosome 6. It has two classes of genes. One class encodes MHC class I molecule that binds cytotoxic T cell through CD8 receptors. Another group of genes encodes an MHC class II molecule that binds helper T cell through CD4. MHC class I gene encodes a protein which are HLA-A, HLA-B, and HLA-C. But MHC class II encodes proteins called HLA-DP, HLA-DQ, and HLA-DR. MHC class I is presented on all nucleated cells throughout the body. But the MHC class II is found on the surface of antigen-presenting cells, such as macrophages and dendritic cells. The second signal is that of co-stimulation. It begins when a ligand called CD28 on the surface of a T cell binds to a ligand called B7 on the surface of the antigen-presenting cell. This region is known which contains TCR, MHC, CD4, CD28, and B7 is known as immune synapse. Once an immune synapse is formed, the naive T cell changes into an activated or effector T cell. After the formation of immune synapse, the antigen-presenting cells send out different cytokines to the cytokine receptor on the surface of the activated T cell. These cytokines help tailor the immune response to a specific pathogen. After that, the activated T cell starts generating a high concentration of interleukin-2 or IL-2. IL-2 is produced by activated T cells and promotes further growth and differentiation of activated T cells. Its effects include the proliferation of cytotoxic T cells, T helper cells, and natural killer cells, while enhancing the secretion of IFN-gamma and TNF-alpha. Interleukin-2 controls CD4 T cell fate by controlling the expression of the key cytokine receptors, transcription factors, chromatin regulators, and effector cytokines. The whole process upregulates the expression of the IL-2 alpha receptor. You may be thinking, why is that? The interleukin-2 receptor has three components: alpha, beta, and gamma. The naive T cell expresses the beta and gamma components of the interleukin-2 receptor, but they have a low affinity for interleukin-2 cytokine in contrast to the alpha component of the interleukin-2 receptor, which has an affinity for interleukin-2 cytokine. Thus, upregulating the expression of alpha IL-2 receptor also increases the affinity of IL-2 cytokine to the activated cell, which in turn increases the production of interleukin-2. This is a form of autocrine stimulation, meaning that the cell stimulates itself. IL-2 cytokines enable the now activated T-lymphocyte to rapidly proliferate to produce large clones of thousands of identical T-lymphocytes. In this way, even though only a few T-lymphocytes in the body may have TCR molecules able to fit a particular epitope, eventually many thousands of cells are produced with the right specificity. This is referred to as clonal expansion. These cells then differentiate into effector T4 lymphocytes and cytotoxic T lymphocytes, or CTL. T-cell activation is not possible without the presence of a T-cell receptor. We have discussed the structure of TCR in detail in a separate video. But let's revise some of the basic points. T-cell receptors are heterodimers, meaning made up of two different types of monomers. These monomers are alpha and beta monomers, while others are gamma and delta monomers. These monomers make T-cell receptors. So one type of T-cell receptor can have alpha and beta monomers, while other types of T-cell receptors can have gamma and delta monomers. Mostly T-cell receptors can have more alpha and beta monomers rather than gamma and delta. T-cell receptors have variable and constant regions. As it is the constant portion of the alpha chain, so it is represented as C-alpha. While this is the constant portion of the beta chain, it is represented as C-beta. The variable region is represented as V-alpha. The variable region or domain of the beta chain, so it is represented as V-beta. This variable region helps the T cell to detect antigens presented by the major histocompatibility complex to the cell receptor. The diversity of TCR is achieved through a process called gene translocation, which in turn is based on combinational and junctional diversity. Both B lymphocytes and T lymphocytes have evolved a unique system of gene splicing called gene translocation, a type of gene shuffling process where various genes along with a chromosome move and join with other genes from the chromosome. T lymphocytes can cut out and splice together different combinations of genes along their chromosomes. Through random gene translocation, any combination of the multiple forms of each gene can join. This is known as combinatorial diversity. As we have seen before, the T-cell receptors, or TCR, of most T lymphocytes involved in adaptive immunity consist of an alpha and a beta chain. There are 70 to 80 genes that code for the variable portion of the chain of the TCR. During gene translocation, specialized enzymes in the T lymphocyte cause splicing inaccuracies, wherein additional nucleotides are added or deleted at the various gene junctions. This change in the nucleotide-base sequence generates even greater diversity, leading to junctional diversity. As a result of combinatorial diversity and junctional diversity, each T lymphocyte can produce a unique shaped T-cell receptor capable of reacting with a complementary shaped peptide bound to an MHC molecule. So, to summarize, the immune system of the body has no idea as to what antigens it may eventually encounter. Therefore, it has evolved a system that possesses the capability of responding to any conceivable antigen. The immune system can do this because, during the early differentiation of naive T lymphocytes in the thymus marrow, each T4 lymphocyte and each T8 lymphocyte becomes genetically programmed to make a T-cell receptor, or TCR, with a unique shape through a series of gene translocations. Molecules of that TCR are put on the surface of that T lymphocyte to function as its epitope receptor. When an antigen encounters the immune system, epitopes from protein antigens bound to MHCI or MHC-Roman2 molecules eventually will react with a naive T4 and T8 lymphocyte with TCR and CD4 or CD8 molecules on its surface that fit. This activates that T lymphocyte. This process is known as clonal selection. That is all about T-cell activation and stimulatory effects that initiate the cell-mediated immune response.